Brain slice preparations has proven to be invaluable for research in neuroscience to shed light on the neuronal electrical properties, screening of diverse therapeutic molecules, and even to study different Alzheimer's disease processes. However, one major drawback is that the physical and biological traumatic changes induced by ex vivo preparations might cause changes in neuronal properties, and therefore, some of the results need to be interpreted cautiously. In this regard, Metabolomics can provide new clues about the whole spectrum of physiological changes, accounting for the ex vivo preparations at different time points. For this study, we have used an integrated and targeted metabolomics strategy employing LCMS and GCMS to expand the metabolome coverage in order to gain a better understanding of the metabolic modifications and the lipid remodeling processes taking place at different time intervals after the collection of brain slices commonly used for the electrophysiological recordings. For metabolites and lipids extraction, just approximately 30 milligrams of hippocampus tissue were homogenized and extracted in one single solvent phase prior to analysis. After univariate and multivariate statistical analysis, 300 compounds were found to be critically affected by the time interval. We detected a clear decrease in critical metabolites of GABAergic synthesis and degradation pathway 2.5 hours after brain slicing, principally GABA, glutamate, and glutamine. Such a reduction of GABA levels would affect the excitation-inhibition balance of the brain circuits. Furthermore, important changes in the levels of pyruvate and lactate have been found 2.5 hours after slicing. Additionally, many intermediates of the TCA cycle also presented temporary alterations in their levels, including citrate, succinate, fumarate, and malate. Finally, we found increased levels of all the glycerophospholipids and sphingolipid species 2.5 hours after slice collection, followed by a sharp decrease, nearly reaching their initial levels after 5.5 hours, as we can observe in Figure 2. The brain slicing itself is a disruptive procedure that shatters the cell membrane through their slicing region. Therefore, it can be hypothesized that the initial increase in the levels of both lipid classes after brain slicing might be related to an upregulation in lipid biosynthesis, including cell membrane rearrangement to reduce the cellular leakage, followed by a stabilization of the tissue within the period between 2.5 and 5.5 hours after slicing. In summary, we have shown for the first time in a preparation used for electrophysiological recordings that complex changes in the metabolome and lipidome of brain slices take place in the cells after the brain slices process. All these preparation-dependent changes in the brain biochemistry related to the time interval should be taken into consideration for future studies to facilitate a non-biased interpretation of the experimental results.